We all know asthma can, well, it can feel like a very personal fight with your own lungs sometimes.
Hmm.
What maybe isn't always clear is just how much inflammation drives that. It's a really complex picture.
It absolutely is.
And today we're diving into one corner of that picture. We're looking at a new potential therapy, AR-RAGE, and how it relates, or maybe doesn't directly relate to a really common marker,
Yeah.
FeNO, fractional exhaled nitric oxide.
Exactly. We've got some recent info and update on FENO data reporting for AR-RAGE trial from May 2025.
Okay.
And also a really interesting piece from LLMJ around the same time, the significance of FENO data in the clinical development of AR-RAGE.
Right.
So our goal today is to really dig into why FENO, which, seems like the standard marker for asthma drugs, might not be the best way to understand what AR-RAGE is doing, or maybe not the most relevant way.
Okay, so FeNO. Lots of people with asthma, or who know someone with asthma, have probably done this test. You just breathe into a machine. What's that breath actually telling us?
Yeah, it's pretty simple for this patient. That breath gives us a glimpse, really, into type 2 inflammation in the airways.
Type 2.
Right.
Okay.
So in certain kinds of asthma, like eosinophilic asthma, specific immune pathways get switched on, and that leads to more nitric oxide being produced. FeNO just measures that nitric oxide.
So high FeNO means?
High FeNO generally points towards that type 2 inflammation being active. It's actually quite useful for diagnosis in those cases and for seeing if treatments like corticosteroids are working, calming things down.
And I guess predicting who might respond well to those steroids.
Exactly. And maybe even flagging a higher risk of flare-ups.
And there are standard levels, right? Like numbers doctors look for.
That's right, yeah. Groups like the American Thoracic Society, ATS, they have guidelines. Generally for adults, under 25 parts per billion, or ppb, is low.
Okay.
25 to 50 is intermediate, and over 50 is considered high.
And for kids?
A bit lower. Under 20 is low, 20 to 35 intermediate, and over 35 is high. It just gives clinicians, a framework.
Got it. So FeNO, clearly important for that type 2 side of asthma. Now, ARO-RAGE, let's talk about what it's targeting. What's its focus in this whole inflammation picture?
Okay, so AR-RAGE is different. It's an inhaled therapy, uses something called RNA interference. Think of it like a very precise dimmer switch for one specific protein.
Okay, a dimmer switch. For what protein?
It targets the receptor for advanced glycation end products. And here's the key difference. RAGE isn't just involved in that type 2 inflammation we were talking about with FeNO. It seems to be a more central hub. It plays a role in both type 2 and non-type 2 inflammation.
So broader than just type 2.
Exactly. It interacts with different signals that promote inflammation, activating pathways inside cells. So ARO-RAGE, by using RNAi, aims to silence the gene that makes RAGE, specifically in the cells lining the lungs.
Reducing the amount of RAGE protein.
Right. Less RAGE protein, and the hope is less of that broader inflammatory response.
Okay, so that sets up the central question then. We've got FeNO looking at type 2 inflammation and ARO-RAGE hitting this broader RAGE target. Why wouldn't FeNO be a great way to measure if ARO-RAGE is working?
Well, it's kind of like trying to figure out if there's a problem with the main power supply to your house by just checking if one specific lamp turns on.
Okay, FL.
FeNO is like that lamp. It mainly shows activity in those type 2 pathways, the ones often driven by signals like IL-4 and IL-13. ARO-RAGE, though, is going after the main circuit board, RAGE, which affects lots of different circuits, not just the one connected to the FeNO lamp.
So ARO-RAGE could be working, maybe dampening down other important inflammatory signals that RAGE controls. But we might not see a big drop in FeNO because that's tied to a different specific pathway.
Precisely. The drug might be having a real clinical benefit or affecting other markers in ways that just don't directly lead to less nitric oxide production measured by FeNO.
Now, you mentioned the sources talk about a trial, ARO-RAGE 1001, and it specifically included patients with high FeNO.
It did, yes. That trial had a specific group, a cohort of asthma patients who started with FeNO levels at or above 35 ppb.
So why do that if FeNO might not be the best marker? What were they hoping to see?
Well, the idea there was to look specifically, does ARO-RAGE have any effect on this well-known marker of type 2 inflammation, especially in people who definitely have that signal elevated at baseline? It's about characterizing the drug fully.
Right, makes sense. So what did they find? What happened to FeNO levels in that high FeNO group?
Well, that's where the public information gets a bit sparse. We know from interim results back in May 2024 that ARO-RAGE did significantly reduce soluble RAGE, sRAGE.
Which means it's hitting the target protein, right?
Exactly. Target engagement confirmed in the overall asthma group. But the specific data on what happened to FeNO levels in that high FeNO subgroup, that hasn't been released publicly, at least not as of May 2025 when these sources were published.
That's interesting, especially because the main study, the primary completion, was March 2025, right? So the data collection should be done.
Right, data collection done, but maybe the analysis of specific biomarker subgroups like FeNO is still ongoing or perhaps being held strategically.
Strategically, what does that mean?
Well, that update on FeNO data reporting source hints at this. It suggests the timing for releasing that specific FeNO data might be tied up in, potential partnership talks or licensing deals. Or it could just require a more complex analysis. Either way, there's definitely interest among researchers and investors to see those FeNO numbers.
Okay, so if FeNO isn't necessarily the star player for measuring ARO-RAGE, what else should we be looking at? What are these potentially better indicators?
Yeah, good question. Several others could give a more direct or complete picture. Soluble RAGE, ARO-RAGE is obviously key.
Because that shows the drug is actually doing its job on the RAGE protein itself?
Exactly. It shows target engagement. And if you see that ARO-RAGE reduction correlating with patients actually feeling better or having better lung function, that's a really strong signal.
Makes sense. What else?
Another one could be HMGB1.
HMGB1.
Yeah, it's a molecule, a pro-inflammatory one, that actually binds to RAGE and activates it. So measuring changes in HMGB1 might show the downstream effect of blocking RAGE, perhaps reflecting impact on pathways FeNO doesn't capture.
Okay, so downstream effects.
Right. And then because RAGE is involved in both type 2 and, importantly, non-type 2 inflammation.
Like neutrophilic inflammation.
Precisely. So looking at markers associated with neutrophils, things like IL-8 or myeloperoxidase, could be really informative about ARO-RAGE's broader impact beyond the typical FeNO-related pathways.
So ARO-RAGE, HMGB1, neutrophilic markers, these all sound much more directly linked to how ARO-RAGE is supposed to work. Which brings us back to FeNO. Why even bother including it so specifically in the trial or reporting on it? Are there good strategic reasons?
There definitely could be. Even if it's not the main story, the FeNO data still adds a piece to the puzzle, right?
Okay.
Seeing what ARO-RAGE does or doesn't do to FeNO in that high baseline group helps build a more complete profile of the drug. How does it interact with different inflammatory aspects?
And FeNO is just, well, no. So gotta interpret FeNO carefully anyway.
Always. Extremely well known in the asthma world. Doctors understand it, researchers expect it, maybe investors too. So reporting something on FeNO might just be, expected due diligence, even if the drug's main strength lies elsewhere.
Sort of ticking a box, maybe?
Potentially, yeah. But also, maybe more than that. There's the chance you might find specific subgroups within those high FeNO patients who do show a FeNO response.
Like responders versus non-responders, even within that group?
Could be. Identifying those potential responders could be really valuable for later trials or for figuring out who might benefit most.
Okay, what else?
Well, think about the competition. Lots of asthma drugs do target type 2 inflammation and lower FeNO, showing how ARO-RAGE compares. Maybe it doesn't lower FeNO much, maybe it does a bit, helps position it. It could highlight its unique angle, maybe for patients who don't have super high FeNO or aren't responding well to T2 biologics.
Right, defining its niche.
Exactly. And finally, even if FeNO isn't your main success measure, the data could still help design better phase 3 trials. Maybe you use it to stratify patients, or just keep it as a secondary outcome to track. And as we mentioned, the timing could be useful in licensing talks.
So lots of potential reasons to keep FeNO in the mix, even if it's not the primary endpoint reflecting the drug's core mechanism?
Precisely. It adds context and potentially valuable secondary information.
And we should also remember, as the LLMJ article points out, that FeNO isn't perfect, right? Other things affect it.
Absolutely. Things like allergies, smoking status, even your diet can nudge FeNO levels up or down, and an individual's FeNO can vary day to day. Plus, a high FeNO doesn't always perfectly match how severe someone's asthma symptoms are.
So, gotta interpret FeNO carefully anyway.
Always. Context is key.
Okay, so wrapping this up then, the big takeaway seems to be FeNO, super useful marker for type 2 asthma inflammation, no doubt about it. But for ARO-RAGE, which targets that broader RAGE pathway involved in both type 2 and non-type 2 stuff, FeNO might not be the most direct or maybe even the most revealing measure of success.
That sums it up well. To really get a handle on ARO-RAGE, We probably need to focus more on those other endpoints that better reflect its mechanism.
Like the ARO-RAGE, HMGB1, maybe those neutrophilic markers.
Exactly. Those are likely to give us a much richer understanding of what the drug is actually doing to the underlying inflammation.
And in the meantime, we're all kind of waiting for that full data package from the ARO-RAGE 1001 trial, including that FENO data from the high baseline group to see how it all fits together.
Indeed. Seeing the biomarker data alongside the clinical outcomes will be very telling.
So it really makes you think, doesn't it? As we learn more about just how tangled these inflammatory pathways are in lung diseases, how might our reliance on single signature biomarkers need to change? Are we heading towards needing more, holistic panels or assessments rather than just looking at one number? Definitely something to ponder. Thanks for digging into this with me.
My pleasure. Fascinating area.